Biochemistry is the chemistry of living matter. We can divide it into organic chemistry and inorganic chemistry. Organic chemistry is the chemistry of hydrocarbons. That is, all of the molecules contain hydrogen and carbon. Organic molecules tend to be large molecules. We call them polymers or macromolecules. They are held together by covalent bonds. Inorganic molecules are very small molecules, relatively speaking. They may contain carbon or they may contain hydrogen, but they do not contain both. If they contain both carbon and hydrogen, no matter of their size, they would be organic molecules. Inorganic molecules are held together by ionic bonds. The exceptions are water and carbon dioxide. They are inorganic molecules, but they're held together with covalent bonds. The inorganic compounds are water, salts, acids, and bases. Water is extremely important to living things because it has some very unique properties. First of all, water has a high heat capacity. It changes temperature very slowly. You know this if you go swimming in a swimming pool. It can have been warm for several days, but the pool water is still pretty cold. And yet, once the pool water warms up, it stays warm for several days after the air temperature has turned cool. Water has a high heat of vaporization. It takes a lot of heat to change water from liquid to gas. Water is as close as we come to a universal solvent. It will dissolve most substances. Water is chemically reactive. We use water in both hydrolysis and dehydration reactions, which are very important in joining together the polymers of organic molecules. And most body cavities have watery fluids in them to act as a cushion and to act as a lubricant. The other inorganic molecules, acids, bases, and salts, can be classified based on what they dissociate into. Salts will dissociate into cations and anions. So sodium chloride is a salt because it dissociates into sodium and chloride. These will conduct electricity, so they are electrolytes. Calcium salts are important in giving our bones strength, and iron salts are involved in transporting oxygen. Acids will dissociate into hydrogen ions and anions. In other words, their cation is always a hydrogen ion. So hydrochloric acid dissociates into hydrogen ions and chloride. The fact that we get hydrogen ions tells us we're dealing with an acid. And bases dissociate into cations and hydroxyl ions. Hydroxyl ions are an oxygen and a hydrogen, but they have an electrical charge. Sodium hydroxide dissociates into sodium ions and hydroxyl ions. Because we get hydroxyl ions, we know we're dealing with a base. Acids and bases change the pH of solutions. So what is pH? Well, pH is defined as the negative logarithm of the hydrogen ion concentration of a solution. That sounds pretty complicated. What do you really need to know? Well, what you really need to know is pH is a scale that runs from 0 to 14, and in the middle we have 7. 7 is neutral. It's neither acidic nor basic. As we move from 7 towards 0, we are becoming more acidic. From 7 to 14, you're going to the basic or the alkaline side. The further you are away from 7 on either end, the stronger you are. So something with a pH of 6 would be a relatively weak acid. Something with a pH of 13 would be a relatively strong base. Now here's a pH scale that shows some fairly common items that you're familiar with. Notice that pure water is neutral, a pH of 7. Seawater, slightly alkaline, has a pH of 8. Wine, tomatoes, pH of 4. Lemon juice is pretty acidic with a pH of 2. Now, before we had pH paper and other ways to tell pH, scientists used to determine the pH sort of in general by taste. Acid things taste sour. Alkaline things are slippery and taste bitter. There are 4 basic organic molecules found in living things. Carbohydrates, which are our primary fuel and energy source. Lipids, which is a fuel storage molecule. This is how we can store energy long term. Proteins, which are involved in building structures in the cell and in performing certain functions in the cell, particularly enzymes. and nucleic acids, which are involved in storing, retrieving, and transmitting the information involved in heredity. Organic compounds are typically polymers, long chains of repeating smaller molecules. These repeating molecules are known as monomers. They are formed by dehydration synthesis. That is, when we want to stick two monomers together, these two molecules have been very happy living independently. We've got to make them want each other, so we have to take something away from each other. We can take a water out from them and we will have them need each other. When we break down these compounds, as we break apart these monomers, they're used to being stuck together. We have to give them something so they don't want to stick back together. So we can release water when this happens. Here you see two monomers, two independent molecules that have no reason to join with each other. If we want to hook them together, if we want to chemically bond them, we have to take something away from each of them. If we take an oxygen and a hydrogen away from this molecule and a hydrogen away from this molecule, we make water. And when we do that, we now have a reason for these two to stick together. We make a chemical bond. This is called dehydration synthesis. We remove water from these two molecules so that they will chemically bond with each other. Now, if we have a larger molecule that we want to break down, if we're going to break this bond, these two molecules are going to want to get back together. We need to give something to heal the break. So if we put water in, when we break the bond, we can give an oxygen and a hydrogen to one molecule and a hydrogen to the other. We now make them independent molecules. This is hydrolysis. This is a decomposition type reaction. Here is an example of the whole reaction. Glucose and fructose can be chemically bound to make sucrose. This is a dehydration synthesis reaction. We take water away from these two molecules, so we get water as a byproduct, and we hook them together to make sucrose. If we want to break sucrose back down into glucose and fructose, we have to put a water in. When we break this bond, we have to use the water to heal up the break so that we can have two independent molecules again. Carbohydrates are composed of the atoms carbon, hydrogen, and oxygen. The monomer, or the building block, is the monosaccharide. A monosaccharide is known as a simple sugar. A polymer is made when we put many of these monomers together in a string. We start out by putting two together. That's a disaccharide. We just saw that with sucrose. As we continue to add monomers to the chain, we have a polysaccharide, a complex sugar or a complex carbohydrate. The primary function of carbohydrates is to provide fuel and energy for our cells. We do have some structural carbohydrates. Plants have cellulose in their cell walls. That's what makes celery crunchy. And insects can have chitin in their exoskeleton. That's what makes the roach crunchy when you step on it. Nutritionally, carbohydrates are things like starch and glycogen. So here are our monosaccharides. These are 6 carbon sugars, and we count each little place here has a carbon in it unless something else is set there. So we have 6 carbons in all of these, and these are 5 carbon sugars. 6 carbon sugars are known as hexoses, 5 carbon sugars as pentoses. As we stick 2 monomers together, we get a dimer, so monosaccharides become disaccharides. We make sucrose, or maltose, which is 2 glucose molecules stuck together, or lactose, which is galactose and glucose stuck together. If we continue adding to the chain, we'll make either starch or glycogen. In this case, we're seeing glycogen. Lipids are composed of the same atoms, carbon, hydrogen, and oxygen. However, here we have a lot of carbon and hydrogen chains, very long chains of nothing but carbon and hydrogen. The monomer for neutral fats is glycerol and 3 fatty acids. The polymers, triglycerides, the neutral fat, glycerol and 3 fatty acids. We can also have phospholipids, steroids, particularly cholesterol, and eicosanoids. Lipids function in long-term energy storage. Also, most of our triglycerides help insulate our body and may help protect us. They cover bony surfaces so that we have some protection. Triglycerides can exist as saturated fats. In saturated fats, there are single covalent bonds between all of the carbon atoms in those chains of fatty acids. It's called saturated because you've got the most hydrogen atoms possible attached to that molecule. Saturated fats are solids at room temperature. They tend to be animal fats, things like butter and lard. Unsaturated fats will have one or more double bonds between the carbon atoms in the fatty acid chains. These are liquid at room temperature, and these are your plant oils. These are typically thought to be heart healthy. Several years ago, when it was decided that saturated fats were very bad for your heart, unsaturated fats wanted to be used. But most people in the United States were used to using butter, and they didn't know how to use a plant oil. So manufacturers started hydrogenating unsaturated fats, that is, adding hydrogen back to them and breaking some of those double bonds so that they would be solid at room temperature. That led to the development of trans fats. These are modified oils where we break some of the double bonds to add hydrogen atoms. It's what makes plant oils solid. This is how margarine was made. However, in recent years, we've discovered that the kinds of bonds that are made when you artificially break those double bonds are very, very unhealthy. Trans fats should be avoided in your diet. So here we see triglycerides. This is a glycerol molecule, 3 carbons in it, and we add 3 long fatty acid chains. These are very long. The little dots here indicate that there's a whole lot more carbons and hydrogens in here. Phospholipids, we take a triglyceride, but we break off one of the fatty acid chains and we add a phosphate group. Now this phosphate group is soluble in water. Fatty acids are not soluble in water. So this gives us a very unique molecule, what we call our polar head, and anything that's polar will dissolve in water, and our nonpolar tails, and they will not dissolve in water. This is a major component of cell membranes. It allows us to have a membrane that can interact with the watery inside or outside of the cell and yet have a water impermeable barrier between the inside and the outside. Cholesterol is used to help stabilize the cell membrane. Here again you have some of these ring compounds, so there are carbons in all of these points. So there's a lot of carbon here. But you can see this is a very stiff, stable kind of molecule. So this can be interwoven in those phospholipid tails and can help stabilize the membrane. Also, cholesterol is a major component of steroid hormones. Eicosanoids are local hormones that are all made from one fatty acid, arachidonic acid. This acid is found in the cell membrane. Eicosanoids are involved in making prostaglandins. This is the most important eicosanoid compound. Prostaglandins play a role in blood clotting, the localized control of blood pressure, inflammation, and labor contractions. The actions of prostaglandins can be blocked by nonsteroidal anti-inflammatory drugs, NSAIDs, and by COX inhibitors. Now, COX stands for cyclooxygenase enzyme. This is an enzyme that is involved in making the prostaglandins that are particularly involved in inflammation. So, COX inhibitors really target inflammatory prostaglandins. Our third molecule is the protein. The atoms involved in proteins are carbon, hydrogen, oxygen, and nitrogen, and some of the amino acids contain sulfur. The monomer is the amino acid, and there are 20 different kinds of amino acids. As we stick 2 amino acids together, we have a dipeptide, and then we have polypeptides. Proteins function to form cellular structures, like elements of the cytoskeleton. They're involved in cellular identity. Receptors on cell surfaces help cells function and identify each other. And they're involved in certain cellular functions. All of our enzymes are proteins, antibodies are proteins, and they provide us with immunity. Proteins function because they have very unique structures. Each particular protein has its own unique structure. We start the protein structure with the primary structure. This is the exact sequence of amino acids. If we change even one amino acid in a chain, we may change the protein. Normal hemoglobin and the hemoglobin involved in sickle cell anemia varies only in one amino acid. As we string together the amino acids, we have some opportunities for parts of the amino acids to interact with each other with hydrogen bonds. This will give us either an alpha helix or a beta pleated sheet, and that is our secondary structure. The alpha helix and the beta pleated sheet put other amino acids closer to each other than they were normally, and so we begin our tertiary structure, more folding based on more hydrogen bonding. Quaternary structure occurs if two or more polypeptide chains react with each other to make the fully functional protein. Protein shape determines its function. We have our primary amino acid sequence. Notice how parts of the amino acid are thrown together so that we can have hydrogen bonding, giving us our alpha helix or our beta pleated sheet. This will put other amino acid groups close together so we can have this more complicated folding. That's our tertiary structure, and two or more held together by something is a quaternary structure. Hemoglobin is an example of a quaternary protein. It's 4 polypeptide chains held together by iron. Every protein has a specific shape, and the function of that protein depends on its specific shape. If we change the shape, we change the function. So how do we alter the shape of a protein that would alter its function? This is called denaturation. Denaturation disrupts bonds, which disrupts the shape. Denaturation may be reversible, that is, it may let the protein fold back like it was, or it may be irreversible. In other words, the protein is permanently damaged. Denaturation can be caused by pH changes, temperature, ionic concentration, or mechanical agitation. Egg white is almost pure protein. And you all know what happens when you cook an egg white. That would be a temperature change. You have denatured that egg white protein by temperature. If you take a little egg white and put it in vinegar, which is an acid, you will see a similar change occur in the egg white after a short period of time. If you've ever beaten egg whites to make meringue, you've used mechanical agitation to begin the denaturation process, and then you have gone on and added sugar or cream of tartar, and that changes the ionic concentration. So you've probably all denatured some proteins at some point in your life. There are two types of proteins. Fibrous proteins are structural. They're strand-like proteins, they're not soluble in water, and they're very stable. They're used to provide mechanical support and tensile strength. These are things like keratin that's found in your skin, elastin that's found in elastic structures, and collagen, which is probably the most common protein found in your entire body. Globular proteins are functional. They're compact molecules that tend to be spherical and dissolve in water. These are the molecules that are most sensitive to denaturation. They have specific functions based on the active region or that particular shape on their molecule that allows them to react with some other molecule. Antibodies, hormones, and enzymes are all globular or functional proteins. Since we're talking about enzymes, remember they're functional globular proteins. All enzymes end in ASE, so you can know you're talking about an enzyme by looking at the word. Enzymes are biological catalysts. Now, in general, catalysts speed up chemical reactions. Biological catalysts are those that are involved in living things. Catalysts are not part of the reaction. They enter and leave the reaction completely unchanged. They simply make the reaction occur faster than it would normally. Enzymes are the catalysts found in living things. Enzymes function by lowering the activation energy of a reaction. All reactions require a little startup energy. This is their activation energy. This is energy that has to be put into the reaction to get it started. Even though the reaction may ultimately give off energy, it needs a little push to get started. So we say that enzymes, if this is how much energy we have to put in to get this reaction going, enzymes level that out. If we have to put in less energy to get the reaction going, the reaction will occur more rapidly. Most enzymes are not always in their functional form. The holoenzyme is the entire enzyme, the functional enzyme. The apoenzyme is the protein portion of the enzyme. This is the part that has the active site, the area that reacts with a substrate. Cofactors, which are metallic ions, or coenzymes, which are organic molecules like vitamins, have to attach to the protein portion to fully activate it. The cofactor and or the coenzyme, some require one, some require the other, some require both, is what alters the shape of the protein and turns it into its active form. Enzymes are substrate specific. The substrate is what the enzyme will interact with. They only act on that one molecule that they are specific for. The substrate has to fit the active site. That substrate and enzyme has to fit together like a puzzle piece or like a key in a lock. So if this is the enzyme that will allow two substrates, let's say these are two amino acids, to be connected together, the amino acids have to fit in the active site. Once they're in there, the reaction putting them together can occur, and the dipeptide leaves the enzyme, and the enzyme is just like it was before we started. So the enzyme made the reaction speed up, but it was not involved in the reaction in any way. Our fourth organic molecule are the nucleic acids. They're composed of the atoms carbon, hydrogen, oxygen, nitrogen, and phosphorus. The monomer of nucleic acids is the nucleotide. A nucleotide is composed of a pentose, either ribose or deoxyribose, phosphate, and a nitrogen-containing base. The polymers are either DNA or RNA. Nucleic acids function in the storage, retrieval, and transmission of hereditary information. DNA is a double-stranded helix. It's found in the nucleus of cells. The nitrogen bases in DNA are adenine, guanine, cytosine, and thymine. DNA sticks together because of complementary base pairing. Adenine always pairs with thymine, and cytosine always pairs with guanine. Hydrogen bonds hold the bases together. During cell division, DNA has to replicate. This allows us to pass on the genetic information to the next generation. Also, it provides the genetic code for making proteins. This is a DNA molecule. The nucleotides are deoxyribose, phosphate, and one of the four possible bases. So this is one nucleotide. Here are four nucleotides held together. Because DNA is double-stranded, two strands connect together. Adenine will always hydrogen bond with thymine. Guanine always hydrogen bonds with cytosine. RNA is single-stranded. Now it's produced in the nucleus, but it functions in the cytoplasm. The nitrogen bases are a little different. We have adenine, guanine, and cytosine just like we had in DNA, but instead of thymine, we have uracil. The RNA functions to copy the genetic code and carry it to the protein production site, which is the ribosome. There are three types of RNA. We have messenger RNA, transfer RNA, and ribosomal RNA, and they all play a role in the manufacture of proteins. Here is your RNA molecule. Again, single-stranded. Your ribose as your sugar, your phosphate, and your base. So this would be one nucleotide of an RNA molecule. Here you have several of them strung together to make a strand of RNA. Adenosine triphosphate, or ATP, is our source of immediate usable energy in the cell. It is an RNA nucleotide with adenine as the base, and instead of one phosphate, it has three phosphates on it. Now, putting together the nucleotide, the adenine, the phosphate, and the ribose is pretty simple. That doesn't take a whole lot of energy. But as you start adding phosphates, it takes additional energy to get that second phosphate on, and then a whole lot of energy to get that third phosphate on. Whenever we store energy in chemical bonds, we can get it back out when we break those bonds. ATP is our energy management molecule. When we take in large amounts of glucose and are breaking it down, it captures that energy. We don't need all of that energy at one time. It can hold that energy until the cell needs it. This is your basic RNA nucleotide: adenine, ribose, and one phosphate. Typical kind of molecule, standard energy to put it together. If we add a second phosphate, that takes a little more energy, And we see this wavy line, that's an indication that there's more energy than usual in this bond. And to add the third phosphate to make ATP takes a lot more energy as well. When we remove the phosphates, we release the energy in this bond. Since there's extra energy in this bond, this is a high energy storage bond. Typically, if we have ADP and some extra energy and phosphate, we can make ATP. We can hold energy and store it for a little while. When we need that energy, we can very quickly break ATP down, get the energy back out. We recapture ADP and phosphate, and we can recycle that when energy is available again. The cell has many uses for ATP. We need it to transport materials across the cell membrane. We need it to do the mechanical work in cells, and it's involved in putting molecules together. We said those end organic reactions required energy, and ATP is going to be our source of that energy.